So Bismillahirrahmanirrahim and welcome back to my channel where I teach pharmacology, pathology and all the subjects that are taught in MBBS. So my name is Ordinary Days, obviously my channel name, and now we are going to talk about adrenoreceptor polymorphism, that is a part of this adrenergic transmission playlist. Now what is adrenoreceptors? As we know that we have a synapse. When we have this synapse, if the substance that is released upon the synapse is a norepinephrine, and the receptor for the norepinephrine is a adrenoreceptor. So adrenoreceptor polymorphism is that these type of polymorphism or these type of structural changes, they are inherited together. That is why they are haplotypes, and they are produced, these polymorphism, different structures, different shapes of the adrenoreceptors, they are produced by changes in critical amino acid sequences. As you know, the receptors, they are proteins. So if we have change in their protein sequences, changes in their amino acid sequences, what will happen? We will get adrenoreceptor polymorphism. Now we will talk about two things in this adrenoreceptor polymorphism. One is receptor regulation and other one is receptor selectivity. So now what is receptor regulation? So as you know, whenever norepinephrine will come to this receptor, it will produce a response, and now this response that they are saying, it is not fixed or a static response. Rather, the magnitude of this response that is produced due to the coming of norepinephrine, it depends upon number and function of the adrenoreceptors and regulation by catecholamines. So it depends upon the number of the adrenoreceptors that are displayed on this postsynaptic membrane, number, and how many of them are functional, and their regulation by catecholamine, other catecholamines that are going to increase the expression of the receptors onto this postsynaptic membrane. So number and function. Secondly, regulation by catecholamines. Now what is desensitization? Desensitization, it is a part of receptor regulation. They say that desensitization is also known by the names tachyphylaxis, refractoriness and tolerance. So what is tolerance or this type? Tolerance is whenever you are exposed to something again and again, you develop a type of you are desensitized to it. You don't think that it's a big thing, rather you say that it's a small thing, so you ignore it. So that is tolerance, that is desensitization. After random or after repeated exposure to the catecholamines, what happens after repeated exposure of the receptor to the norepinephrine? What will happen? The receptor will now produce less response. It will become less responsive to further stimulation by that agent. And what happens? It decreases the therapeutic response, or it decreases the type of the response that is to be produced, because now this receptor is less responsive to this type of agonist. Why? Because it has been recurrently exposed to it again and again, again and again. So now how this desensitization or tachyphylaxis or tolerance or refractoriness, how does this develop? We have certain mechanisms for it. So the mechanism for desensitization, it can be a slow one, which goes up for like hours or days, and it involves transcription and translation. Anything that is slow, it is going to involve genes in it, and genes are rather regulated by transcription and translation. And a fast one that is occurring in minutes, it involves covalent modification and changes in subcellular locations. So in covalent modification, either you are going to attach a phosphorus group, that is a phosphorylation of the receptor, or you will have a second messenger feedback. And then changes in subcellular location, the second thing, it can be that the receptors, they go inside the postsynaptic membrane, so that is subcellular location. So up till now we have studied desensitization, which is less response because of repeated exposure, and now we are studying the two mechanism. Either it can be slow one by involving transcription and translation, and either it can be a fast one, and fast one has covalent modification, either attachment of a phosphate group to that receptor, we attach the phosphate group, either we regulate it by second messenger system, or either we change its subcellular location. Now categories of this receptor, we need to know about these categories. They are homologous and they are heterologous. The categories of this tachyphylaxis or desensitization. What is homologous desensitization? They say that if this is the receptor, the main receptor, So this agonist or this norepinephrine is coming to it and the receptor is being exposed to it again and again. What will happen? Repeated exposure to the agonist will develop a type of sensitivity, and this is homologous one. But what happens in heterologous one, that this receptor, it is exposed to this agonist, but now not only this receptor is affected, but another receptor which is not exposed to the agonist, it is not being repeatedly exposed to the agonist, but because this receptor has been desensitized, the other receptor also desensitizes. So this is type of heterologous desensitization. Homologous is receptor, you can say homologous is direct one, whereas the heterologous is indirect one. Another receptor is being desensitized, and that will be responsible for desensitization of some other receptor that is not being repeatedly exposed. So homologous direct, heterologous indirect type of desensitization or inactivation. Now we will talk about the fast mechanism of desensitization, which involves phosphorylation of receptors. So they are saying that this phosphorylation, generally what happens that whenever you have this adrenoreceptor, whenever it is exposed to the agonist, it is going to help or work by a G protein coupled receptor kinase. So it is going to exert its function by this G protein coupled receptor kinase. And adrenoreceptors provide substrate for this receptor. So what happens is that this is the receptor and the agonist is the substrate, and this substrate to the G protein coupled receptor kinase is provided by this adrenoreceptor. So this is the whole mechanism. Now this is homologous desensitization, because you can see what is happening. The agonist is coming and it is directly attaching to the receptor and it is directly inactivating it. But now what happens? That now we have attached a type of we have done the phosphorylation of this adrenoreceptor that was coupled to this G protein regulatory kinase. What will happen due to this attachment of phosphoryl group? This attachment of phosphoryl group will enhance the affinity for a substance which is the arrestin. So now a substance which is arrestin will now attach to this adrenoreceptor instead of the agonist. Now arrestin substance will attach. This arrestin substance will remove the G regulatory kinase and it will do the G protein independent signaling pathway. And what it will do, it will block the G protein by steric hindrance. It is going to alter its structure in such a way that G protein regulatory kinase is now blocked, and arrestin has evoked such a response that now this pathway is not going to occur, rather a G protein independent pathway is going to occur. And this arrestin has the capability to start the clathrin and clathrin adapter AP2 type of proteins, and what will they do? They will endocytose the receptor. So up till now what have you understood? That normally this receptor is bound to G protein regulatory kinase. Okay, it is doing its function, and when it is repeatedly exposed, what will it do? It will develop desensitization. But what happens by phosphorylation is that now we are going to endocytose that receptor. We are going to remove it from the postsynaptic membrane to the inside, to the cytoplasm of that postsynaptic membrane. And this will be done by, firstly, if there was this receptor onto the postsynaptic membrane, we will attach a phosphate to it. So due to this attachment of phosphate, now instead of the agonist, the receptor is going to bind to this arrestin. This arrestin will activate clathrin and clathrin adapter AP2 proteins. They will do endocytosis of the receptor. Secondly, it will deactivate this G protein pathway that was attached to it. So two things: activate clathrin and remove the steric hindrance of the G protein pathway. So now this receptor will be doing its action independent of the G protein, and it will be endocytosed, and it will not be displayed onto this postsynaptic membrane, and because of this we will develop less number of receptors onto the postsynaptic membrane, and hence desensitization will take place. So the major chunk that you can get from it is that phosphorylation of receptors will lead to their endocytosis. Major chunk is this, and this will be a type of homologous desensitization. Now we will talk about second messenger feedback. Now second messenger feedback is from the category of heterologous desensitization. So what will happen generally is that, for example, we have the beta adrenoreceptor. That beta adrenoreceptor is related to this G protein pathway, and that is a G stimulatory protein pathway. And whenever G protein stimulatory pathway is there, what happens? Amount of cyclic AMP increases. This cyclic AMP activates protein kinase A, and this protein kinase A in turn will do the phosphorylation of the receptors. So it is a bit enhanced process, and that phosphorylation will now what it will do? It will lead to endocytosis. We have studied in the previous one. Now in beta 2 receptors, whenever this agonist is repeatedly coming, what will it do? It will activate the GS pathway. It will increase cyclic AMP, activation of protein kinase A. Now what will happen? There will be phosphorylation, but on the third loop of the G protein pathway or the G protein receptor. So onto the third loop, cytoplasmic loop of the beta 2 receptor, a phosphate group will be attached on its serine residue. Now what will happen? Instead of being associated with the G stimulatory protein, it will now be associated with the G inhibitory portion, and it will decrease the amount of cytoplasmic AMP that was actually being produced. So this is how conformational change is being produced by this phosphorylation. In the other step, the other phosphorylation that we studied by homologous one, what was that? It was involving endocytosis. But in this one, what it is going, it is changing its affinity, and now it is not affinized, or now it is not having affinity for GS, rather it is having affinity for GI, and hence the major pathway will be inhibited and cyclic AMP will be decreased. Now this type of also what happens, the beta 2 receptors, they have the capability, they can either be attached to GQ pathway, and GQ pathway can activate protein kinase C. GS activates protein kinase A, and GQ activates protein kinase C. The protein kinase C also does beta 2 receptor phosphorylation similar to this one, and then this it activates this G inhibitory pathway and decreases cyclic AMP. Now either the beta 2 receptor is getting desensitized by the protein kinase A activation or protein kinase C activation. Both of them will, what will they do? They will phosphorylate this beta 2 receptor. They will attach a phosphate group to it. But not only they are going to phosphorylate this beta 2 receptor, apart from that, all the receptors in the cell that are similar to this beta 2 receptor, they will also be phosphorylated. Phosphate group will be attached to them by these two proteins. And what will happen in such a way? That agonist was repeatedly being introduced directly to one substrate, one beta 2 receptor, but this activation, that was internal cellular activation of protein kinase A, protein kinase C, it affected receptors that are similar to it. They were not actively exposed to it, rather they indirectly got phosphorylated. So this is heterologous desensitization, indirect desensitization. So I hope you understood. Phosphorylation of the receptors and second messenger feedback pathway, both of them, they occur in a few minutes. And what do they lead to? They lead to desensitization within very small span of time. So we are done with this receptor regulation, that was desensitivity of the receptor. Next thing we are going to talk about is receptor selectivity. Now they are saying that selectivity is like a specific receptor shows response to a specific type of substrate. But now the selectivity is not absolute. If the selectivity is absolute, absolute means that if it only responds to one single chemical, that will be known as specificity. But selectivity is not absolute. It can show response to the related agents. So now they're saying that drug which activates one receptor at a concentration on which it will have no effect on another receptor at the same concentration. So what it is saying that, for example, there is a particular type of drug which is activating beta 1 receptors. For example, it is beta 1 selective. So it is given in a particular concentration like 5 milligrams. Now it is going to activate beta 1 receptors on that type of dose, but when it goes to the beta 2 receptor, it has no effect on that receptor. So this is the type of receptor selectivity that takes place. Now clinical effects, what happens? Selectivity to adrenoreceptor and relative expression of receptor subtypes in given tissue. So because of these clinical effects, because of the selectivity, what will happen, that only a type of receptor that is selective for the type of drug will show the response, and also it depends upon the amount of the receptors that are available. For example, if beta 1 are more in number and beta 2 are less in number, so the relative expression will be more on the beta 1 as compared to the beta 2. So relative expression is important, as well as selectivity to the receptor is very much important. Now what happens, we did an observation that whenever there is this receptor which is attached to this G protein kinase pathway, it can either function by G protein kinase pathway or G protein independent pathway. So it has both the capabilities. So they observed this thing and they developed drugs which are biased agonist. Biased agonists are drugs that have the capability, for example, if there is this agonist, it can activate only this pathway. So it will be a biased agonist for protein coupled pathway, GPR, G protein coupled pathway. And if we do not want the G protein pathway to be activated, we do not want cyclic AMP to be used, what will happen, we will develop a biased agonist that will activate this G protein independent pathway. So this observation led to the development of drugs that can be classified on the basis of their pathways. So this is biased agonist. And what are allosteric modulators? So what is, when you studied enzymes, you have talked about allosteric site. Allosteric site is the site apart from the main site where a type of substance, when acts, it will change the conformation of the receptor. So what happens in allosteric modulation? Now, for example, this is an agonist, it is binding to the normal site, the agonist binding site, or agonist attachment site. Now, secondly, what is going to happen, that an allosteric modulator will come, it will attach to the allosteric site and it will produce a conformational change. Now, this conformational change can either lead to increased attachment to the agonist, or it can diminish the receptor in such a way that receptor cannot bind to the agonist. So this modulation is done by allosteric type of agonist. So we have done about receptor selectivity as well, that a particular drug which is effective for a particular receptor might not be effective at that concentration for another type of receptor, and that is selectivity. And that selectivity helped us to develop biased agonist, and that selectivity can be influenced by allosteric modulation. So up till now we are done with the whole mechanism of adrenoreceptor polymorphism. So you have talked about receptor regulation and you have talked about receptor selectivity. Both of these things have been clearly explained. We have explained desensitization, that was part of receptor regulation, type of responses, that is phosphorylation of the receptor that leads to endocytosis or second messenger feedback pathway that can not influence the endocytosis, rather it can influence the selectivity, or it can do, what can it do, it can cause the G inhibitory pathway to be activated, decrease cyclic AMP, or it can lead to heterolocus desensitization. And what is the receptor selectivity? That we can develop due to this mechanism, we can develop biased agonists that are that can activate one particular pathway, and they have therapeutic effects for that pathway. And this selectivity can be differentiated, can be modulated by allosteric modulators. So I hope you are very much fine with it. We are done with it. Now let's move on to the next portion. If you like my videos, please like, share and subscribe, and keep on watching Ordinary Days. Allah Hafiz.